Hey, so we're back with another lecture, and what this lecture is going to talk about is just the very basic first stuff that I think I missed in the last talk about the cell bio intro. So this flip lecture is based on what Stein talked about for the first thing that she talked about for cell bio, which is the cell bio intro. I think it's important that we know this too because I didn't talk about this in the last lecture. It's just one page, not a lot of things to talk about, and it's just pretty much facts from this point. So pretty basic stuff. The Earth is 4.6 billion years old. So basically what happened was dust particles started to gather after the Big Bang, and within 3.5 billion years the origin of life occurred. So the very first thing we talked about the domains of the tree of life, eukaryote, archaea, bacteria. So the main man that started to, so even before there was DNA, there was RNA, and the man that studied RNA was Voice. I think that's how you say it. It's Woese. So that's him. So he studied ribosomal RNA, and that pretty much changed everything. So it changes along the branches, and it changes the rRNA. So there's difference. So we also talked about the differences between eucs and procs. Obviously, procs are single-celled organisms. They have a cell wall. They usually occur within archaea and bacteria. And the main difference, even though eucs and procs have flagella, the flagella in eucs is made of microtubules. So the number one difference that I guess we talked about is that eucs have nucleus and the procs have the nucleoid that stores all their genetic material. And another thing is eucs have membrane-bound organelles, and the procs just have ribosomes and DNA and all of that. So what's the difference between plant cells and animal cells? Plant cells have cell walls, they have chloroplast, while the animal cells have nothing like that. But the plant cells do have mitochondria. That's the one thing that animal cells have, and the mitochondria helps to generate ATP from sugars. That's the same thing that the eucs do in animal cells. So that's one thing. So the flagella in prokaryotes is made of flagellin. It's a protein. Pili is made for sensation. And it's also stored in capsules, which is protection for sugars and carbs. So there's many ways that Stein talked about and how we can measure these things and how we can look at cells. And one was electron microscopy, which is more powerful than light microscopy. And so basically what that happens is electrons hit nuclei of atoms and heavy metals bind to proteins and all of that. So now we can talk about the plasma membrane of prokaryotes. The plasma membrane of prokaryotes is made of a lipid bilayer, and that bilayer is filled with proteins, and the empty space inside of each bacteria is the nucleoid. So usually in these pictures, the dark spot is electron dense, and the light spot is electron lucent, which is what you talk about. So that part has all the lipids and nucleic acids and the fats and all of that. So again, we talked about how single-celled organisms are protists. Fungi is diatoms, plants is the pea leaf stomata, yeast is budding yeast, and mammalians is obviously us. So in the nucleus, our DNA is stored in the nucleus. 10 times bigger than procs. Cytoskeleton may be found in procs, but usually it's just in us. Membrane-bound organelles just in us. So what is the universal feature of living organisms? Obviously, they have hereditary material. They have DNA. They copy and replicate. Some DNA turns into RNA, that's transcription. The RNA turns into proteins, and the proteins catalyze and enzymes, and that's translation. And they require free energy, and all cells have plasma membrane, right? So what's the main? So what are the? And we can end just this video on what the 5 things are that all living organisms have. So cells have a plasma membrane. They have genetic material for hereditary use, energy, evolution, and replication. So just remember those 3 things, and you'll be fine. That's it for today.